engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology. Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to the Naked Scientists podcast. This is the programme where we bring you the biggest breakthroughs in science, technology and medicine. I'm Chris Smith, and this week, Titans of Science continues, and joining me is vaccine specialist Andrew Pollard, one of the architects of the Oxford AstraZeneca COVID jab.
From Cambridge University's Institute of Continuing Education, this is The Naked Scientists.
Andrew Pollard was born in Tunbridge Wells in Kent on the 29th of August 1965. He attended St Peter's Catholic School in Bournemouth before studying medicine at St Bartholomew's Hospital Medical School in London. He trained in paediatrics at Birmingham Children's Hospital, specialised in paediatric infectious diseases at St Mary's in London, and then joined British Columbia Children's Hospital in Vancouver as their clinical fellow. He currently serves as the Ashall Professor of Infection and Immunity at the University of Oxford and is Director of the Oxford Vaccine Group at the Department of Paediatrics. He's chaired the UK's Joint Committee on Vaccination and Immunisation, the JCVI, about whom we heard an enormous amount during the COVID pandemic, since 2013, and he's also a member of the World Health Organisation's Technical Advisory Group on salmonella vaccines. Andrew's played a pivotal role in the development of numerous vaccines across his career, including those for typhoid, meningitis, influenza and Ebola. He was knighted in the Birthday Honours List in 2021 for services to public health, particularly during the COVID-19 pandemic. Andrew, a very warm welcome to Titans of Science.
Thank you very much, and thanks for having me.
That's quite a CV. Why did you end up going down the infectious diseases route and the vaccine route in the first place?
Well, I actually got into the vaccine route later. I started off really interested in the immune system, and particularly the immune system of children. If you go back 250, 300 years, half of all children died during childhood before the age of 15. So 1 in 2 children died. Most of those died from infectious diseases. So there's no world in which having lots of bad things happen to you and surviving them is the right thing to do. You really need to have an environment around children that protects them from these life-threatening illnesses. And we've achieved that through public health since really the mid-1800s, when acts around clean water and air pollution started to appear, which then started a trend of plummeting under 5 mortality in this country that really was finished off by the advent of vaccines and at a large scale in the 20th century.
Do we have any insights then into roughly what the scale of morbidity and mortality is that are being prevented by vaccines? How game-changing they really have been worldwide?
Well, if we think about the annual lives saved in childhood from the global vaccine programs, which are not as comprehensive as ours here, we're talking about somewhere between 3 and 5 million lives saved every single year. So yes, a huge impact. And if you look at it in the context of individual diseases, we have smallpox, which was a terrible pandemic which went on for centuries, and it wiped out millions and millions of people. And that is a virus that's completely eradicated from the planet. If we think about measles, which is a virus which, when I started in pediatrics, was killing 1 million children every year, the global rollout of vaccines over that 35-year period, which is still not reaching everyone, but it means that we now have reduced measles mortality globally by about 90%. The impact is just mind-blowing. When I started out in pediatrics in the early 1990s, there are diseases I saw regularly then, which my trainees now here in Oxford today just do not see at all. And I find that incredibly invigorating and exciting to have been involved in vaccination over that period of time that's just wiped out the experience of junior doctors of seeing these awful diseases.
How do vaccines actually work, though? What's the simple kind of mechanism by which what Edward Jenner did a few hundred years ago translates into protection from an infectious disease?
Well, essentially, whether you're Edward Jenner or the latest generation of RNA vaccines that were used in the pandemic, they're all doing the same thing, which is training our immune system to recognize things which are foreign on the surface of bacteria and viruses, but doing it in a safe way. So you give either part of a virus, or you train the cells in our body to make parts of a virus or a bacterium, so that our immune system can be educated, so that when we actually meet the real bug in the wild, the immune system already knows what to do and gets rid of it, rather than meeting it for the first time, the infection taking seat in our bodies, causing disease and killing children before we've had time for our immune system to be educated.
They fall into different categories, though, vaccines, don't they? In some cases, we actually give live vaccines. In other cases, we give killed vaccines. In other cases, we give bits of the infectious entity that's been busted apart, chemically brutalized. And in other cases, we give toxins that they make. So what sort of determines what we use and how it works?
I mean, some of it is because we knew the mechanisms of the disease related to particular components. So you mentioned the toxins. One great example of that is tetanus toxoid, which is one of the vaccines we give routinely in 5 doses through childhood. And tetanus is a bug which we find in the soil, and it releases a toxin after it gets into wounds in our skin, which can cause paralysis. And it's just by making an immune response against the toxin that we're able to stop the disease. We're not fighting the bug. For other viruses, we know that you can just take one part of the surface of the virus and use that. So the shingles vaccine, which we now use in the UK in the elderly, just has a protein that sits on the surface of the shingles virus, which is the same as the chickenpox virus. And that boosts our immune responses so that we're able to keep shingles at bay. For other vaccines, people have taken traditional approaches of saying, well, what if we weaken a virus like measles? Could that then, without causing any significant disease, protect people from measles? And by trial and error, many vaccines were tested many decades ago to show that this worked. So in many ways, we choose the different type of vaccine, partly just on history and partly because of some scientific knowledge about which is the right bit of the organism, the bacteria or the virus, to target.
We saw this manifest during the pandemic, that with certain vaccinations, it's a one-stop shop. You have either the disease or, ideally, you get vaccinated against that disease and you get 1 or 2 doses of that, and you never get that thing for the rest of your life. On the other hand, we were updating COVID vaccines almost by the month, it felt, during the pandemic. So why is there that distinction? Some things it's a lifelong protective effect. Other cases you need to change the vaccine.
Well, it's a slightly complicated issue. If we take something like coronavirus or influenza, we need to have regular vaccines if we want to completely prevent infection with them, because the viruses are changing all the time and we can never quite keep up with them. And if through mutation the virus changes, the vaccines that we had last year are no longer as effective against them. It doesn't mean they're not effective at all, but they're not as effective because the viruses, as it's growing, it makes mistakes all the time in making new copies of itself. And some of those new copies completely evade the immune responses that we've already made. And so that means that you can get reinfected with coronavirus over and over again, even though you've been vaccinated. The important thing, though, with COVID-19 is that the vaccines still today that we had back in 2021 are vaccines which stop you dying, but they're no longer very good at stopping you from getting an infection. And those infections can still be pretty miserable. But now, if you meet a coronavirus, you don't get that overwhelming disease, which we saw with our intensive care units full of very sick patients, because the vaccines are still providing good immunity against that severe endpoint. With other viruses, they really don't change very much. and so survive by causing infections in unvaccinated people. And they spread if they're unvaccinated people. And measles is a good example of that. The measles vaccine still works brilliantly today, just as it did when it was first used in 1960, because measles virus has not really changed very much over that period of time. But if you allow a pool of people who are unvaccinated to be in a community, as happens in many countries where there's poor vaccination coverage, then the virus can still sustain transmission amongst those individuals and cause explosive outbreaks. But once you've been vaccinated, you're protected for life because you've got the immunity against the virus and it doesn't change very much.
And presumably, if you have enough people in a population who are vaccinated, that's where this concept of herd immunity comes in, because there are so few people left who can still catch the infection that the chances of an infected person running into a non-immune person are so slight, you just can't get a chain of transmission.
That's correct. So you don't get a chain of transmission, but for that unimmunized individual, if they did meet someone with measles infection, they could get infected. And it's so infectious, it's highly likely that they would do. And the reason for maintaining very high levels of immunity in the population is to stop those chains of transmission happening, both because it's potentially fatal for children and adults who get measles, but also because there are some people in our community who cannot be vaccinated because their immune system doesn't work properly, they're on cancer treatment, and those people really need everyone around them to be building that shield of immunity to keep the measles virus out.
How do you decide what we're going to vaccinate against? Because there must be umpteen things that we could theoretically protect everybody against, but we decide on a certain suite of things we're going to give routine paediatric vaccination against, and then we let people catch other stuff. So how does that decision get made?
Well, the starting point for decision-making and trying to develop vaccines over the centuries has been a focus on the very severe diseases, those that cause death. And most of those diseases in childhood that are relevant in the UK that happen on any scale, we do now have vaccines for. It doesn't mean that we can prevent every infectious death, but we have done incredibly well over, particularly the last 60 years, but really going back into the early 20th century when the first vaccines were starting to be used on a larger scale. So that's really the starting point. But today, now that we've really dealt with those really big killers like diphtheria and measles and so on, that we've got vaccines for them, the focus is much more on how can we make sure that what we're doing in adding any intervention here in the UK for the National Health Service is good value for taxpayers. And the way that that's determined is by a metric that's set by the government's treasury around cost effectiveness. And what that means is that we have a level playing field. So if we're going to introduce a new vaccine, it should have the same prioritization economically as any other treatment the NHS decides to introduce, so that we don't end up using a vaccine to prevent some mild illness at the expense of introducing a new treatment for cancer that would have improved much more of the quality of life across the population. And if we just take the health service perspective, you can do pretty exquisite calculations around all the different types of medicines and vaccines that we could use in the NHS to only prioritize those where we meet those threshold for being good value for the taxpayer.
And is that the role of the JCVI in the UK, the Joint Committee on Vaccination and Immunisation, which you chair? Is that committee's role to weigh up those things and make recommendations?
It is absolutely. So, the starting point is actually, do we have a burden of the disease? We have really good data in the UK from the NHS, which is collated by the UK Health Security Agency, about a whole range of diseases that affect the population. The second thing is there a vaccine? And that is academic and commercial developers who are working on products that they hope to be able to then market in populations. And so we then need to look at the data and scrutinize about whether the vaccine does work. Is it safe? what is the potential impact in the population? And then lastly, that economic arbiter of does it meet the Treasury rules on cost effectiveness?
All this must have been like a massive dose of steroids, though, once COVID came, because the committee must have been used to making decisions at a much slower pace. Not often that new vaccines come along and have to be evaluated. And so all of a sudden you were confronted with making decisions about a whole new disease, whole new groups of the population who might need to be protected, with a whole new slew of different vaccines all coming along all at once.
Well, the first thing to say is that the committee does review new vaccines every year. It's the bread and butter. We're often reevaluating the current schedule and perhaps reduce the number of doses because new evidence emerges that protection is sufficient. And sometimes we find that you need extra doses, and the wonderful surveillance system we have in the UK allows us to do that. When it came to the pandemic, I was working on the Oxford AstraZeneca vaccine and the clinical development of it. And so I actually stepped back from chairing the COVID committee for JCVI in the pandemic. And my colleague, Wei Shen Lim, took over that role in order to then make those decisions. And they really did work very hard, meeting often several times a week to review that huge deluge of data and new information that was being provided.
But we got the impression, I mean, I'm talking in terms of the general public seeing this, we got the impression that the government were asking committees like the JCVI to make really important decisions about things like, well, do we give COVID jabs to kids? And sort of almost justifying what the political position was going to be based on what the JCVI was saying. But you were operating with very, very limited data.
Well, as I say, I wasn't involved myself directly in that decision making, but I think one really important thing in decision making around policy in such a situation is that you have to make decisions. The data are almost always incomplete, but if you do nothing, it results in people dying. So the decision making has to be the decisions made on the best evidence available at the time. And always with programs like this in an emergency, you're constantly reviewing and revising your decision making, depending on new information as it comes.
Did we learn a huge amount, though, about how to conduct trials at scale, do it very quickly and make decisions very quickly? Or did we go too fast, do you think? Because it really surprised everyone when all this started. And we were told, well, normally it takes years to get a new drug even out of a test tube and into a person's arm, and then to do all the trials and to get the approvals and so on. And a decade's work was effectively condensed into, 10 years became 10 months almost, didn't it? So what have been the learning points to come out of what happened during the pandemic, which have now improved and streamlined how we do this sort of thing now?
I would look at it slightly differently. I would say that we knew a lot about coronaviruses. And because there had been small outbreaks of coronaviruses over the 20 years prior to the COVID-19 pandemic, we already knew how to make the vaccine. There was an outbreak of the original SARS virus, severe acute respiratory syndrome virus, in 2001, and then the Middle East respiratory syndrome virus in around 2012. And those were pretty awful infections. The first one killed about 10% of those infected, and the second epidemic about 30%. So that really focused the minds of trying to understand how to make a vaccine for coronavirus, so that when 2020 came, we actually didn't have to do decades of research because we already knew exactly what to do to make a vaccine. And all we had to do really was that last sprint to the finishing line over that 9 to 11 month period in order to then have a vaccine for licensure. So the only bit we were doing was the clinical development to show that in a large number of people, in our case about 25,000, that the vaccine was safe and it was efficacious. The reason why that bit could be done quickly was simply that you didn't have to wait for someone to pay for each of the next steps as you went through the phases of clinical development, because we had the money provided, the clinical trials. I say provided, but we did have to go and beg for it to run the clinical trials and get us all the way through to having the data to submit to regulators for them to scrutinize. Whereas that normally takes many years, just because each time you do one stage, you then can spend a year or two trying to find someone to pay for the next bit of the development.
The vaccine that you helped to develop, the Oxford vaccine that then became more widely known as the Oxford AstraZeneca vaccine, that actually is a sort of backbone technology that has been redeployed in various directions, hasn't it? Because you've used it in a range of different disease contexts. Ebola benefited from that backbone, didn't it, as well? Am I right in saying that?
Yes, I mean it's the viral vector. This is adenoviruses. One of the Ebola vaccines uses a different adenovirus, an adenovirus type 26, which is the product produced by Janssen or Johnson and Johnson, who's now the parent company. We used a slightly different adenovirus from chimpanzees called Chadox for our backbone for our trials.
But basically, to make a long story short, it's a virus that acts as a Trojan horse. It takes into the body a bit of the coronavirus that you want to persuade the immune system to recognize.
Well, it takes actually the gene from a bit of the coronavirus, and then our cells make the bit of the coronavirus, so that we're using really our cells as a factory to generate the vaccine, which we then make an immune response against.
How did AstraZeneca get involved? Because you had that technology, you were already working on that. So how did it end up being the AstraZeneca-Oxford tie-up?
Well, what we know how to do in Oxford is to design vaccines and to do clinical development. But we're not a global pharmaceutical company, we're a university. And so what we really needed was a partner who had expertise in manufacturing and distribution on a massive scale. And what AstraZeneca did was they brought in incredible expertise in that global space that allowed them to actually take on board over 20 manufacturing sites to try and protect the manufacturing across regions of the world from nationalism, and also to then distribute those doses to the countries and geographies where the vaccine was needed.
It was done at cost, though, as well, wasn't it? That was another attraction. Or was that at the insistence of your team in Oxford, that they do this at a scale that isn't going to end up breaking the bank for countries that otherwise might not be able to afford vaccines?
Well, AstraZeneca was chosen as the partner because they were prepared to distribute the vaccine not for profit. And for the university, this was very important in a pandemic, that there was no sort of perverse incentive to sell just to rich countries. But actually, because there's no profit, it doesn't really matter whether you're going to low-income settings, middle-income countries, or high-income countries. The point of it is to make sure it gets to those who are at the greatest risk, which, of course, are the oldest in any of those populations.
What about the risk to the company, though? Because is AstraZeneca, in 2020 I'm talking, are they, to their mind, taking a big risk, taking on the technology in an untried, untested sort of way, apart from obviously the initial trials that have been done? Are they worried about their reputation? And were there conversations around what could happen and what safeguards need to be put in place to protect the reputation of you at Oxford University, of AstraZeneca and the public?
Well, I think the discussions really were about in taking this on were, first of all, there was a strong view from the leadership at AstraZeneca that there was a moral reason why they should be doing this, because they had some capabilities which we didn't have. And it was an opportunity to provide those skills in the pandemic for the population. I mean, there's clearly also an important point for any pharmaceutical company that if they are able to do that, there's a commercial advantage in that you contribute to ending a pandemic earlier, which is very disruptive to any business. So there's a good argument for using the skills and the abilities you have within the company to try to get the world back on an even keel quicker, to then be able to get back to the main job of the business.
What was it like, though, to watch as something that you've developed is going out on million scale dosage levels around the world, and effectively it is changing the course of the pandemic, and did I mean, you saved millions of lives effectively, didn't you? I mean, what did that feel like? Was it both scary but exhilarating at the same time?
Well, it's a really interesting question because, I mean, eventually there were over 3 billion doses produced, and some analyses suggest that over 6 million lives were saved in 2021 alone from the Oxford vaccine. But it's very difficult to connect with what that actually means. And it's always a problem for vaccination that people who don't have a disease and don't die don't realize that they've benefited from it. And I think to some extent, when you're doing, as I do, working in vaccine development in the lab and in the clinical trials area, the pandemic wasn't so different in terms of the work that we did to feel that it was somehow in a different world. What was different was the intensity, without any doubt, and the public scrutiny. But in many ways, it's hard to connect the work that we did, which is just using our normal ethos about what we're here for, how that connects to actually the impact that there is. And I think I didn't feel personally this sort of sense of some amazing achievement, but there were moments like when, in giving interviews in public arenas and so on about it, and people just came up in droves saying, "Thank you, you saved my life." And that to me was very moving when those moments happened. But I certainly didn't sit every evening thinking how clever I was, because that's not really the way in which one approaches these things. It's much more a technical and logistical challenge that you're facing each day.
There were some people who suffered some severe side effects, though, weren't there? People who developed brain blood clots. Did we get to the bottom of why that happened and why it happened to the people it did happen to?
That's right. So with fortunately a very rare event with the first dose of this type of vaccines, it wasn't just the Oxford vaccine, there was also another adenovirus vaccine which was developed by Johnson & Johnson, which had exactly the same side effect. And it's a rare disorder where individuals can develop clots in some parts of the body, but unusually associated with a very low level of what are called platelets, which are components in the blood which are important in helping clotting. So it's sort of almost the opposite of clotting. And that combination of effects led individuals, very sadly in some cases, to die as a result of the event. That condition, we understand there's an association. There's some odd things like there seem to be some quite different rates of that happening depending on which country you look in. It's been more common in Scandinavia, a bit less common in the UK, and rare in Southern Europe. We don't really understand why that is, but I think there's a general view that there is some unknown reason for population differences in the rates of the disease. But we still haven't got to the bottom of why it occurs. There's lots of theories out there, but none of them are proven, and they're actually really difficult to test, because to test them, you'd really like to have an animal model where animals reliably develop the same condition, and then you could try and change the vaccine and see if you could stop it from happening. But we are not able to do that. And so there's this theory, but we don't have an answer of what caused this rare condition.
You must have been very excited, though, when we did usher in the mRNA vaccines, because of the enormous impact that had, not just on the pandemic, but on the potential for application in other aspects of medicine.
Well, I think there's two things about the RNA vaccines which I think are terrifically exciting. The first is just how quickly you can make an RNA vaccine at scale. So if there was another coronavirus pandemic, and you had all of the necessary supply chain in place, you could manufacture millions of doses of a new vaccine within 6 weeks. And if we look at traditional approaches to development of vaccines, the manufacturing bit from a standing start, you may be in the thousands of doses in 3 months. But actually, to get to the millions or billions of doses is a really tall order. But now that manufacturing for RNA vaccines is more mature, I think the potential to be at millions of doses in a short period is incredible. It's a sea change in the potential for fighting pandemics as a result of that. And the reason is that RNA vaccines are essentially, it's a chemical process to make a new vaccine. Whereas most of the vaccines that we have at the moment for routine programs all over the world are complex biological products. They're made by growing something, purifying something from something that's been grown. And that really is a much more complicated, slow process, much more difficult to control. And for that reason, it makes RNA very exciting. The other thing with RNA vaccines is the potential to iterate, where you can very easily make many different RNA vaccines in the laboratory and test them to see which one works best before you go into studies in humans, or you could even make multiple vaccines for humans to find which one works the best. And with that in mind, with one of the areas I work on with plague vaccines, we've made large numbers of plague vaccines over the last couple of years using RNA technology. And that just is almost impossible to do with other types of platform.
Titan of Science Professor Sir Andrew Pollard from the University of Oxford. Now, next time, we're going to be delving into the mysterious realm of the fungus. Where did these organisms come from? How are they changing in response to climate change? And what's their relationship with us and the other animals and plants that we share the planet with? The Naked Scientist comes to you from the University of Cambridge's Institute of Continuing Education. It is supported by Rolls-Royce. I'm Chris Smith, and from all of us here at the Naked Scientist team, thanks for listening, and until next time, goodbye.